Oh, you recorded it. Okay, well done. Thanks, Marco. Does everyone see that?
Yeah.
Okay, great. So hello, everybody, and it's really a pleasure today to welcome Rinke van Tatenhove-Pel from the University of Delft, from the TU Delft, to our seminar today. And she's going to tell us about population dynamics in spatially structured environments. And I have a small biography here. So Rinke did her master's in life science and technology in Delft and a PhD at the in Amsterdam before moving to Delft, where she's in industrial microbiology. And she's done beautiful work on how microbes grow in compartmentalized environments and how they evolve, and also understanding interactions between cells and high throughput screening and selection systems, as well as working with synthetic consortia, predictive models, and lab experiments to improve biotechnology. And so it's going to be really nice to hear from her today. So I will stop sharing. Maybe you can share, Rinke.
Yes.
Oh, I forgot to say that Rinke is happy to be interrupted. So if anybody has a question, you can either just kind of turn off the mute and let rip, or you can write it in the chat and I will keep an eye on the chat and interrupt if a question comes up.
Yeah. I don't know whether I can see when, because I of course see my own presentation mode, so I don't know whether I can see when there are questions, but indeed, please.
I'll keep an eye on it.
Yeah, perfect. Well, thank you, Rosalind, for your introduction and also for the opportunity to present here. So, yeah, when I was asked to speak in this seminar, I thought, well, what should I talk about? And then I thought, well, population dynamics. Well, my expertise is also spatially structured environments. So let's talk about population dynamics in spatially structured environments. And here you see two examples of the spatially structured environments that I work with. So on the one side, you see a polydisperse water in oil emulsion. And on the other side, a monodisperse water in oil emulsion. So these are small water droplets surrounded by oil. Either of different sizes or of the same size for the poly and the monodisperse ones. And we can actually put cells inside these droplets and grow them in this way in a spatially structured environment and compare that to growth in non-structured environments. So that's what we will look at today. So to start, why are we actually interested in spatially structured environments at all? Well, if we look at how organisms grow, and here we see an example, a microscope picture of the yogurt consortium. So that's Lactobacillus bulgaricus and Streptococcus thermophilus. And these strains have a cross-feeding interaction. And when they are grown together, you see that the thermophilus guys only grow well when they are close to this very long bulgaricus guys. But if we replace Streptococcus thermophilus by another lactic acid bacterium that is not depending on Bulgaricus for its growth, we see a completely different way in which these organisms grow. So now the Lactococcus chromoris strains, they just grow everywhere and they are not depending on Bulgaricus anymore. So this shows that spatial structure actually affects the way that microbes grow, and the way that microbes interact with each other also affect, again, the way they form a spatial structure. So I think this is really interesting to try to understand what is going on in these systems and how it affects how the organisms interact with their environment and with each other. So I will talk about three different subtopics today. And the first one is about laboratory evolution in spatially structured versus spatially non-structured environments. So first, what do I mean with laboratory evolution? So when we have a certain population of cells and we apply a certain mutagenesis, or it can also be random mutations coming up. Let's say we now have one mutant out of 4 in total that has a certain property that we are interested in. What we then do in laboratory evolution is that we apply a certain selective condition. So you can compare it to a sieve, and this selective condition should ensure that the phenotype of interest produces most offspring, such that after going through the sieve, after going through these selective conditions, we have more of the mutants that we are interested in. So for example, now every other strain is this mutant. And if we then propagate this multiple times, so have this grow the system in these selective conditions multiple times, in the end, we can enrich for a strain with the property we are interested in. So what does that look like if we do this in actual experiments in the lab? So normally when we do experiments in the lab, we just have one bulk solution where our cells are growing. So we have a pre-culture, we grow our cells. Let's say we have one fast growing mutant and two wild types. We put them together in our medium, we let them grow. And what typically happens here is that the faster growing mutant outcompetes the wild type, because this one, the faster growing guy, consumes more of the nutrients, it propagates faster, makes more offspring, and therefore it's enriched over time. So if we do this multiple times, we also call that serial propagation. So we take again cells in the end, we incubate them again and again. In the end, we enrich for our fast growing mutant. So serial propagation in non-structured environments typically enriches for high growth rates. So those are the mutants that you find in these systems. But high growth rates is not always the thing we are interested in. So one of the questions we asked ourselves is, what actually happens if the sieve that we look at, so this selective pressure, the selective environment, is a spatially structured environment instead of this non-structured environment where we always find these fast growers. So let's have a look at what we can just think that would happen. So here we have the system we normally have. I will also call that a suspension. So that's a non-structured environment. We have our cells, we have the substrate that is surrounding the cells, and basically every cell has access to all the substrate that is there. So there is competition between the different cells. But if we generate a structured environment, for example, by making these water in oil emulsions that I showed in the beginning, so we now have small water droplets. We have our cell in there, and each droplet now contains a pool of substrate which is only available to this specific cell. So each droplet acts as a separate batch culture, we say. So as a separate culture, this cell only has access to this substrate, this cell only has access to that substrate. So there is no competition on substrate consumption between these different compartments. So if we use this system, well, what does it then look like and what do we select for? So we have our cells. Now my Zoom window is in front of my slide. So I can minimize it, I think, or not. Yes, this is better. Okay, so we have our pre-culture. We have our cells. We encapsulate them inside these droplets. And here you can see a microscope picture of what that looks like for Saccharomyces cerevisiae. So we have a Saccharomyces cerevisiae cell inside this water droplet. If we then incubate the systems, the cells can grow inside these droplets. And now, as I said, there is no competition on substrate consumption rate between these different droplets. So let's say here we have a wild type cell that grows very fast, but can make only 2 daughter cells. So now we will have in total 3 cells here. But the green cell is a mutant. It grows way slower, but it is able to make more offspring. So this is now able, this cell is able to make 4 cells in total. So what will happen if we propagate the system? So we break the emulsion. That means we separate again the water and the oil phase. We have a water phase with our wild type and our mutants in there, which is now enriched for these high yield mutants. And we have an oil phase. And if we repeat this several times, again, serial propagation, we enrich for the cell that actually has a high cell number yield on substrate. So basically we enrich for the cell that makes most offspring, because if you make more offspring in the next round, you will inoculate more compartments, will be able to grow more, and in the next round again inoculate more compartments. So you will in the end win the competition. So this was our hypothesis, that this would happen if we would have a spatial structure, but of course we wanted to test this with experiments. So this was done by me together with Emiel Zwering at the VU in Amsterdam. So what we did is we started with a proof of concept experiment. I always like to start with proof of concepts because you already know upfront what is expected to come out, and you can also test your experimental system. So what we did is we made a mixture. 80% of the cells were wild types, and these wild types, Cerevisiae strains, they have a high growth rate and a low biomass yield. And we had 20% of a so-called Crabtree negative mutant. This is a mutant that respires instead of ferments. Doesn't really matter for what that exactly means if you don't know. But these strains typically have a lower growth rate, but a higher biomass yield. And this kind of trade-off is something we see more often. So typically cells that are efficient grow slow, and cells that are inefficient grow fast. So we had this mixture, and what we did is we propagated it both in a spatially structured environment and in a non-structured environment. And what we saw is that in suspension, so in the non-structured environment, indeed, the wild type strain with the fast growth rate is enriched. We also predicted how fast this enrichment would be, just based, we know the growth rates of the wild type and the mutant, so we could predict if we mix these two together, how fast will the wild type outcompete the mutant, and that is this solid line. And the dots are the experimental data, so you see it fits quite well. We can quite well predict how fast the wild type will outcompete the mutant. And if we do the same, also these propagations, but now we do it in these emulsions, now we see that actually indeed this Crabtree negative mutant, so this high yield mutant, even though it has a lower growth rate, it still enriches and it outcompetes the wild type. And here again, the solid lines are the predictions on how fast we think the wild type would outcompete the mutant based on how many offspring we know they make. And also there it fits quite well. So if we grow organisms in a spatially structured environment, we can enrich for cells that have a low growth rate, but a high biomass yield. Well, then the next step was to do this with let's say a bit more challenging condition, where we do not have a proof of concept where we know what we put in, but where we just do a mutagenesis. So we took a Saccharomyces cerevisiae strain, we mutagenized it, and then we propagated it 21 times, both in emulsion and in suspension. So this is approximately 170 generations. And this is what we saw in time. So on the x-axis, you see the propagation number. And on the y-axis, you see the cell concentration that we measured after 48 hours, so after each propagation. And you see that, well, first, both of them increase, both in suspension and in emulsion. But in the end, in emulsion, we find the highest cell concentrations, higher than what we find in suspension. And we took these strains and we sequenced them, and we consistently found mutations in the hexokinase 2 gene in these emulsion evolved isolates. So what did we do? Well, that's actually something we started and picked up here in Delft with Sakarika, PhD, my first PhD student here. I'm still messing up with the Zoom thingy. So. Yes. So what we did is we looked at these hexokinase 2 mutations and we reverse engineered those. So what we did is we took a wild type and we took one of the, we actually took all of them, but I show the data for one of those, of the mutations that we found in hexokinase 2 and reverse engineered that into the wild type and then characterized both the wild type and the mutant. And what we saw is that the growth rate of the hexokinase 2 mutated version of the strain went down, so from 0.4 to 0.33. So indeed, we have a lower growth rate. If we look at the biomass yield, so that now here is gram dry weight per gram glucose, so gram dry weight produced per gram glucose consumed, we see that this really went up a lot. So the wild type was 0.12. That's typically the biomass yield you find in a fermenting Cerevisiae strain. But in the mutant, we found 0.25 gram dry weight per gram glucose. And we also looked at the cell number yield. So this is kind of the number of cells that you can make per picomole of glucose. To get some reasonable numbers out of it, we use picomole here. And you see that that one is increased even more. And this is because if we go back to the selection system, so there are multiple ways in which you can kind of become dominant in this system. So you can make more biomass, so increase your biomass yield. That's what we were hoping for, so to make more grams of biomass per gram glucose. But you could, of course, also make yourself smaller. So instead of having one big cell, if you can make two small cells in the next round, you will also you will be in two compartments instead of in one. So making your cell size smaller also gives you an advantage in this system. So that's also why we say actually the selection pressure in this system is on the amount of offspring that you make. And this offspring you can increase either by increasing the amount of biomass that you make or by decreasing your cell size. And that's something that we saw. So we saw both an increase in the biomass yields in terms of gram dry weight per gram glucose, but also we saw an increase in the cell number yield, so the number of cells made per picomole of glucose. And we also indeed saw that cells became smaller. So this showed that indeed high biomass yield and high cell number yield mutants can be enriched when we grow in spatially structured environments. All right, so that will, I see a question.
Hi there. Just to clarify this cell number point, I guess, I mean, if your cell yield, if the yield is increasing, I can make more cells per glucose. So I don't necessarily follow that the size should get smaller. Have you also looked at the size?
Yes, we looked at the size. So you can see it, it's a bit hidden here, maybe. You can see that the biomass yield, let's say, doubled, but cell number yield more than doubled. So this means that the increase in cell number yield can not only be explained by the increase in biomass yield, and we know from other data that the cells indeed also became smaller. So that part of it is also caused by that.
Okay, all right, thanks a lot. So you could also see it microscopically, basically on the microscope or something.
Yeah, it was not that pronounced, but if you you can kind of calculate size by dividing your dry weight by your number of cells. So then you get kind of the weight per cell. And we saw that that went down. We also tried to measure them on a cell sizer, but the one we had in our lab was not working that well. And the one we had in another lab showed some weird data. So this is actually something we still need to look into, but because it's not really visible under the microscope, maybe if you start measuring stuff, but I mean, there are dedicated machines to do this. We only need to find one that works.
All right. Thanks a lot for clarification.
Yeah,
great.
Thanks for your question. All right. So this first example showed that population dynamics are different if you are growing in a structured environment compared to when you are in a non-structured environment. So certain phenotypes are beneficial in a non-structured environment, and other phenotypes might be beneficial in a structured environment. So the second topic I want to talk about is to now bring it a step further and to look at microbial communities. So these now we have one cell competing with another cell, but they are both of the same species, the same strain, maybe with a mutation in there. But what happens if we look at consortia and what do they do? And that's also inspired by, again, these figures where we saw that if there is cooperation between microorganisms, we see different behavior in a spatially structured environment than when there is no interaction between these cells. So what happens if we look at cooperative interactions in these systems? So first, to look at this question, what we started with is to design a synthetic consortium. So as I already said, I like proof of concept systems. I like systems that I can kind of upfront try to understand. So we designed our own synthetic consortium. And this was made of Lactococcus crumoris strains. You might hear me say Lactococcus lactis every now and then. It's they recently changed the name of one of my favorite pet organisms. It's very inconvenient, I can tell you, but it's the same, Lactococcus crumoris and Lactococcus lactis. So we had two cooperators, and the first cooperator is shown here. It consumes glucose as a carbon source, and it expresses a protease that cuts casein into peptides and amino acids. And then the amino acids can be taken up as nitrogen source. And we added another strain, cooperator B, and this strain consumes lactose. Lactose is a disaccharide consisting of glucose and galactose. And it consumes the galactose to grow, but the glucose it cannot use. It lacks all the glucokinases. So the glucose is secreted. But this guy cannot use casein. So basically we now have a system where cooperator A depends on cooperator B for its carbon source, and where cooperator B depends on cooperator A for its nitrogen source. So they only grow well when they are together, if we add lactose and casein to the medium. Then we also added a cheater, and this cheater consumes lactose, it consumes glucose, and it consumes amino acids. So it's basically eating everything that's there, but it doesn't contribute anything to the cooperation. And if we take these three guys and we culture them together, what we see is that the cheater is enriched in a non-structured environment. So this is just by propagating it in normal, for Chromoris, you don't need shake flasks, we just do it in tubes. If you propagate it in tubes, what you see is that over time the cheater is enriched, but the cheater cannot grow in the presence of lactose and casein and on itself. So basically the whole population collapses, the cell numbers go down, and the whole thing basically dies out. And that is because this cheater actually does consume everything that is there, but doesn't contribute. So it has an advantage, but in the end, it also kills the whole system. So that's what happens in a non-structured environment. And this is actually, I really like this. I think it's very interesting to see because we do see these kind of cooperative behaviors. For example, in the yogurt consortium that I showed, and probably Andreas knows much more about it than I do. But here, we do see organisms that are actually cooperating. But how is it then possible that these systems are stable? How are these cooperation interactions evolving when, if all of the guys are together, the cheaters in the end win? So how does cooperation evolve? So when we we're thinking about this, and it's also known in the field, it's that spatial structure can play a big role here. So if we look at our consortium and at the different combinations of strains that we can make, so as I already said, only when we have cooperator A and cooperator B together, we have good growth. And for all the other ones, they are basically not growing because they either lack a nitrogen source or they lack a carbon source. So then we thought, I was sick last week, I'm still having a bit of a cough. We thought, what would happen if we would put this consortium in our spatially structured environment, in our water and oil emulsions? So if we visualize it, if we start with a 1:1:1 ratio of the different strains, we add them to the droplets. So we can actually decide, and that's a bit of a sidestep now, we are, as experimenters, we can decide how we distribute these cells over the droplets. So this follows a Poisson distribution. It's basically just having boxes and balls, and the Poisson distribution describes how these balls are divided over the boxes if you mix them together. And that's basically what we are doing as well. We have droplets, we have cells, and the ratio between the amount of cells that we add and the amount of droplets that we make, that determines how many cells per droplet we get. So in this case, we used a lambda where we have sometimes 1 cell per droplet and sometimes 2, but we try to limit as much as possible the having 3 cells per droplet, because we know if we have 3 cells per droplet, the cheaters will outcompete the cooperators. So that's what we want to minimize. If we then take the system, incubate it, only when we have A and B together, we will have good growth. In all the other compartments, we will have no growth. So that means that if we break the emulsion, we will now have enriched for these cooperators, because only they could grow. And if we repeat this again multiple times, we should be able to enrich for the subcommunity that produces most offspring. So in the previous case, we selected for the mutant or the cell that makes most offspring, but now by having them together, having a small consortia, we enrich for the combination of cells that produces most offspring. So again, this was a nice idea, but of course we needed to test it in the lab, and that's something that I did together with a master student at the time, with Anjani. And what we did is actually, we took this, basically did this. So we took the consortium, placed it in droplets, and propagated it in time. And what we saw is that in this spatially structured environment, indeed, we lose out on the cheaters, and we increase the number of cooperators in time. So serial propagation in emulsion can indeed enrich for these cooperators. And the solid lines again are model predictions based on how the cells are divided over the droplets. So this Poisson distribution, that we know how the cells, how much offspring they will make if they are together. And in that way, we can predict also what this enrichment will look like. And it worked out quite well. So this, I think, was really cool that we could show it. But we also realized that the system we looked at now was quite simple. In a way, it was really on or off. So they don't grow at all when they are on their own. When they are together, they boom, grow to the max. So we thought, well, probably it's more realistic to have a bit of cooperation independent background growth. That's what we call it. So what we mean with that is that the cells, when they are on their own, they can grow a little bit, and when they are together, they can grow a lot. And this, for example, also holds for the yogurt consortium. When they are on their own, they can grow to some extent, but if they are together, then they can harness the full potential of the medium. So we were wondering, okay, if we have such a more realistic system, what is going to happen? Will it still work? And here I worked together with a mathematician also working at the VU at the time, Daan de Groot. And we looked at different lambda values, so at different numbers of cells per droplet, to see what would happen to the cooperators and the cheaters. And what we saw were basically 3 different regimes. So I think the easiest one to understand is this one. So if we have a high lambda value of 7, we have on average 7 cells per droplet. That means that in most droplets, we will have at least 3 cells or more. And we know that if we have all 3 cells together, so both the cooperators and the cheaters, that the cheaters outcompete the cooperators. And that's also what we see here. If the lambda becomes very high, so if we have a lot of cells per droplet, then the cheaters are outcompete the cooperators. On the other hand, we also see that if the lambda is very low, so that means we have a lot of droplets. That's actually the example as I showed it before, where we had so a lot of droplets with 1 or 2 cells, but very little with 3. If the lambda is very low, then we saw that also the cheaters outcompete the cooperators. And that was first a bit surprising to us. But then we started to think about it more. And that's, I think, always the beauty of models. That it really encourages you to think about your system and to find things that feel maybe counterintuitive, but actually if you start thinking about it, you think, ah, yeah, of course, it makes sense. Because what happens if we have very little cells per droplet on average, we have a lot of droplets that contain 1 cell, and we have only a couple of droplets that contain 2 cells, and even less that contain 3. But that was also our aim. We didn't want to have 3 cells per droplet because then the cheaters would win. But what happens here is that if the cells on their own can grow a little bit, so let's say we started with 25 cooperator A, 25% cooperator B, 50% of the cheater, as it is drawn here. We let the system grow, and now the cheaters on their own, they can grow a little bit. So let's say up to 3 cells per droplet. And when we have the cooperators together, they can grow up to 8 cells per droplet. And you would then say, okay, so that means the cooperators will win. But actually, if you count and you pull them again, you see that the cheaters outcompete indeed the cooperators. And that is because we simply have way more compartments that contain cells that can grow a little bit. And it's very rare to have a compartment where the cells can grow a lot. So I always try to think of it in a way, if we have 1 compartment where cells can grow up to 10, but we have at the same time 10 compartments where the cells can grow up to 2 on their own, then still these guys that can grow on their own outcompete the ones that cooperate. So this is what explains at least what happens at these low lambda values. You simply have too many compartments where the cells can grow a little bit, and that still allows them to outcompete the cooperators. And well, the model then also predicted that there is a regime in between where so an average lambda value of 2 where the cooperators can outcompete the cheaters. So well, this is all modeling again. We wanted to test it in an experiment. So what we did is we added a little bit of amino acids to our medium. And this small amount of amino acids allows the cheater and also cooperator B to grow a little bit. So this is this background growth that I also showed here. So they can grow a little bit when they are on their own, and they can grow to the full capacity when they are together. And we did this both at a low lambda of 0.15 and at a high lambda. And this indeed confirmed the model results that if the lambda is too low, we lose our cooperators. but if we pick a higher lambda, we actually enrich for cooperation again. So what we concluded from this is that cooperator fitness requires intermediate founder population sizes, so intermediate numbers of cells to start with, so not too little and not too much. And this is why we also called it this fly a middle course, Icarus. I don't know if you're familiar with this Greek story, but it basically means that if you're a guy that made wings for himself, if he would fly too high, the sun would melt the glue and he would fall in the sea. If he would fly too low, the water would hit and he would also become too heavy and drown. But if he would fly in the middle, then it works. So that holds here for these consortia as well. If they're too high, the cheaters are out competing. If they are too low, there are just too many compartments that contain only a cheater. And the chance of meeting your cooperating partner is just too rare. So that also doesn't work. But if you are in the middle, then you can enrich for cooperation. So that was this second part. Just taking a quick look to see if there are any questions on this part. If not, then I will continue with the last part. Because then we also wanted to...
Oh, can I ask you a question?
Oh, yeah, sure. Yes.
Yeah, so when you go to lambda, like the 3 or 4, what happens if the cheater and the two cooperators are simultaneously present in a droplet?
Then the cheater will still grow, outcompete the cooperators within that droplet. So
yeah, that makes sense.
Yeah, because that's what we also saw when we tried it. So this is what will happen basically. So in the droplets where we have them all three, the cheaters will win. But still having this higher lambda just to increase the chance of having compartments with only cooperator and cooperator A and cooperator B together is already gives the benefit to the cooperators.
I see. Great. Thank you.
I have a question. Is there a way that you can track what happens in individual droplets? Like whether a droplet that really contains only one cell at the beginning does better and stuff like that?
We didn't do that. So, but some people use microscopy for it. So they label the different cells with a fluorescent label, and then they actually track what's happening in the individual droplets. So it is possible. We didn't do it. You can also use fact sorting, and I will come back to that in the next story from a slightly different angle. But let me see, I want to have this slide. We were also thinking if you can use, I don't know if you're familiar with fact sorting. So what you can do is you can measure each droplet and you get a fluorescence and a scatter signal for it. And basically you can actually see, well, this droplet contains only little cells, this droplet contains a lot of cells. And then you can say only keep the ones that have a lot of cells. So using fact sorting, you could, for example, selectively pick these guys, and you could also get rid of the cheaters. But that's of course nature. I don't know. Then you need to think of a way. How could nature have a way of kind of selectively picking these guys? Because if it's just stochastic, if it's just no interference, let's say from some kind of selective pressure here, then it will collapse. But yeah, you can track it, but we didn't do it.
Okay, thanks.
Yeah.
Can I ask a question?
Yep.
So already at, let's say, lambda equal to 2, I would expect that if the propagation is done more frequently, the cheaters would come more into contact with the cooperators. So, and then this would lead to a decline in the cooperators. Is that something that you would observe from the model?
Yes. So what we saw, I have to remember, I think it is, I think in the supplementaries of the manuscript, is that actually this optimal lambda depends on the frequency of your cooperators and your cheaters. So if you have really low cooperators frequencies, then the optimal lambda is a bit higher because want to increase the chance of them meeting more, let's say, if you already have quite some cooperators together, then it's better to have a lower lambda. And the model results here are for all kinds of different combinations of different initial fractions. So probably these lines at the bottom are cases, now I have to say it right, no, the lines at the top are cases where we already have a lot of cheaters. And the lines at the bottom are the ones where we have, where you want to be even higher maybe than 2. Do you get what I mean?
Yes, but I think so. I was more thinking about the frequency of the propagation.
Frequency of the propagation. Yeah, but that's kind of depending on each other, because if you propagate,
Yeah.
you also change the frequency.
Yes. Yeah.
So it's if you want to do it optimally for each propagation, you would have to pick a different lambda, let's say.
Okay. Thanks.
Yeah. But in general, indeed, if you increase the number of propagations, you will increase the number of cooperators. So having more propagations, you can go to a lower lambda. But we indeed have some data on this as well. But in nature, or even in experimental systems, it's hard to know where you are up front, of course. So you don't always know your frequency of your cooperators and your cheaters. So you have to pick something that works most of the time.
Right. Yes. Thanks.
All right. So that brings us to the last part where we, so this is work of Sagarika, PhD student. And we again work together with Dan, who is now working in Zurich. And what we now were wondering is, can we predict microbial interactions using these spatially structured environments? So now we saw that, okay, depending on the interaction, we see a different spatial structure. So does that also mean that by looking at how the cells grow in a spatially structured environment, that we can predict whether we are actually in this case or in this case, so where we do have an interaction or where we do not have an interaction? So I think this is a really cool question, because it would be very cool if we can just take a sample and predict what the microbial interactions will be. So now this is done a lot with just looking at genomics and saying, okay, this gene is present, this gene is present. So probably if these guys are together, they can work together. But if we could actually measure it and predict it, I think that would be really, really cool. So what we did is, the idea here was to take again a consortium. We have our initial cell fractions, put them in these droplets, incubate them, and now depending on which cells are together and depending on the interaction between them, we get certain growth. So in this case, for example, A and B stimulate each other. So now in both of them grow, while if they are on their own, they do not grow. And here, only this guy is growing in the presence of this guy. So this guy is apparently kind of helping this strain, just as an example. And now we went to fact sorting. So as I just explained, with fact sorting, we can measure for each of these droplets what is in there. And we say, okay, only keep the ones that have a lot of cells, and the other ones we throw away. And for the ones that we keep, we can then determine the final cell fractions. So we have initial cell fractions, we distribute them over droplets, we grow them, fact sort them, and then we have our final cell fractions. Oh yeah, exactly this. So initial fractions, we have our lambda that describes how the cells are divided over the droplets. And we have the final fractions. And now we put that into a model. And the idea is that out of this model, we then get our interaction terms. So, well, what does this model then look like? And I think that will also make it a bit more clear what the rationale is behind it. So we have our initial fractions and we have our lambda. our lambda gives the probability of beads that we have that have 1 cell or 2 cells or 3 cells, etc. So this is basically the Poisson distribution, describing how many droplets have a certain amount of cells. And combined with the initial fractions, this gives us the probability of having different combinations. So the chance of having only an A, the chance of having an A and a B together, chance of having an A and a C together. This depends on 2 things. It depends on the fraction of A, B, and C we had in the beginning, and the number of droplets with 1, 2, or 3 cells. So we have a Poisson distribution and we have a multinomial distribution. And then this probability of the different combinations combined with certain interaction terms will give us then the final fractions. So yeah, maybe I can go back here just to make it clear. So we have our initial fractions, the ratios here. Then we know the number of droplets that have 1 cell, the number of droplets that have 2 cells, 3, 4. We have then certain interaction terms that describe how the cells are going to interact and whether they are going to grow. And then we select for the growing ones and determine the final cell fractions. So what are then these interaction terms? We have 2 of them. So I'm still used to calling it CIJ, but we changed it to GIJ. I might sometimes still say CIJ. And FIJ. And this GIJ, that's actually the number of cells that we get per droplet. So it is relative. And it tells you whether we have a lot of cells per droplet or very little cells per droplet. And the Fij gives us the fraction of cells within the droplet. So this will have a Gij of close to 0, because this we set as a reference as 1 when the droplet is full. So this will have a Gij of 1, and the fraction of both cells will be 50-50. And this will have a low Gij because there is only little growth. This one we will have again a high Gij, but now we will have a high fraction of this cell type compared to that cell type. So that's how we describe the interaction. How well can the cells grow when they are together? So what is the cell concentration that they reach? And within this cell concentration, what are the fractions of the cells that are present? So the model can then calculate these final fractions based on these interaction terms. And we also experimentally determined these final fractions. And then, of course, we can let the model iterate, change these in these interaction terms to find what are the most likely interaction terms that we find. And then the question is, can we decipher all the pairwise interactions in the system in this way? So we took the same consortium as I showed you before. So now we call it strain A, B, and C. The colors are slightly different, but the organisms are the same. We again have this guy that that consumes glucose and makes the amino acids. We have the guy that consumes lactose, secretes the glucose, and needs the amino acids. And we have the guy we before called cheater that consumes amino acids, glucose, and lactose. So that's the consortium we worked with. And we used a lambda of 0.3. And at this lambda value, most of the filled, the beads that are filled, so we also have quite some empty beads, but the beads that are filled, most of them contain 1 cell, a couple of them contain 2 cells, and we have very little droplets with 3 or more cells. So we again look for the pairwise interactions. So we maximize the number of droplets with 2 cells. And minimizing the number of droplets with 3 or more cells. So what we did is we did the experiment, as I said. So we had different initial fractions. So here we had 40% of strain A, 20% of strain B, 40% of strain C. We incubated them, grew them, sorted them, and determined the final fractions. And we did that for 3 different initial conditions. So here we had 10% of A, 30% of B, and 60% of C. And here another initial condition. And for all 3 initial ratios, we determined the final ratios. And this sounds like, this looks like a simple plot, but this is actually quite a lot of work to do all the growth and the fact sorting, determining the fractions, and so on. So this is takes, if everything goes well, generating this data takes 6 weeks, I think. And yeah, as you know, things almost never go well the first time. So quite some time investment here. But if we then take these initial fractions, the lambda and the final fractions, and put it into the model, this is what we got out. So the G for A and B, so this is set as the reference. So this will always be 1. And what we see is that when A and C are together, there is no growth. And when B and C are together, there is growth. And if we then look at the fractions, so if we have A and B together, we have 39% A and 61% B. While if we have B and C together, we have way more C and only 26% B. So what does this now mean? So this means that if we have A and B, they both grow. If we have A and C, there is no growth, so there's no interaction between these 2. If we have B and C together, then basically mostly C is growing. And yeah, we know that actually B is not growing because we start with a certain amount of cells, which is close to 23%. So this 26%, those are basically the Bs that were present already from the start. So B is not growing, but apparently it is feeding C. So then we wanted to check with our experiments whether this is indeed the case. So we verified these model results in suspension cultures. And there we saw indeed that when we have A and B together, they grow. B and C together also grows. A and C together, they don't grow. And A and B, we see that we have, well, close to equal contribution of both of them, where for B and C together, we here basically only see the C's. So let me see. So this shows that we can predict the pairwise interactions, which I think is quite cool. But yeah, we also thought, well, this is only one consortium. So if we go back to the consortium, we can also see what is happening. So if we have A and B together, that's the same as the 2 cooperators together we had before. So this guy secretes the glucose, which is consumed by this guy, and this guy provides a nitrogen source to that guy. So they grow together to a 50-50 ratio. But we also saw that when we have B and C together, only C is growing. And what we think is happening there is that we have a certain amount of B present in the beginning. It hydrolyzes the casein to make these amino acids. These amino acids can be used by this strain together with lactose to grow. And even though this strain is not providing any glucose back to strain B, the amount of amino acids that are released by strain B is enough for this strain to grow to the full capacity of the beet. So that was what was happening here. But then we thought, yeah, I mean, we actually need to show it with more than just one consortium. So we took a different strain C here, C star. So A and B are still the same, but now C star is not able to use the lactose. So it can only use glucose. And this means that we now have a different interaction between B and C, because B is now still releasing the amino acids, but C star cannot use the amino acids because it doesn't have a carbon source available. So we think, okay, we now have, for this consortium, we knew that these two could grow together. These two, only C would grow. We take another consortium where we think that C will not be able to grow, and we again try to infer the interactions. So what we did is we again had several initial fractions of strain A, B, C star, and strain A, B, and C. And what we did is we fixed the interaction between A and B and A and C in this case. So we only looked at the interaction between B and C. So that means in the model, we just tell the model A and B together grow, A and C together do not grow. And now tell us what is happening between B and C and between B and C star. And what the model showed is that when we have B and C together, there is growth. So A and B again was the reference, was 1. So we have a bit less growth here with B and C than we have A and B together. And we mainly see C's. So this is the same as what we saw before when we predicted all 3 interactions. And the model also predicted that when we have B and C star together, so that is the second consortium, then there is no growth. And when we checked that again with suspension cultures, we saw that indeed B and C, as I already showed before, we only have C's that are growing, and B and C star, we indeed see that there is no growth. So again, we see that we can predict pairwise interactions in this way, which I think is quite cool. So this is all the work of Sagarika. It's the first time for me to present it. I hope it was clear, this last part. But if there are questions, I'm happy to take them. So these are the people that I want to thank. I see, well, we still have quite some time for questions. I only used half an hour, but that's fine, I think. Or no, an hour, right? It's, oh, so I talked a lot. Well, we have 2 minutes for questions. These are the people that were involved. I showed most of them along the way. These are all the sponsors for the different projects. And I want to thank you again for this opportunity and for your attendance.
Thank you very much, Rinke. That was a super nice talk, really clear. Thank you very much. I'm sure there's a lot of questions. I actually have one question before I have to go in 2 minutes, but Marco is going to take over. So hopefully there'll be time. Maybe I should ask my question first and then hand over to Marco. My question was just about what happens if you have a more complex community? Is there a way that you can use the droplets to extract like interaction parameters then?
Yeah, so that's one of the things we are looking now. So we are simulating data for more complex communities with more strains. And what we see are different things. So if we go to consortia, let's say of 6, 7, 9 different strains that are present, the number of experiments you need to do, of course, increases also a lot because you simply have more parameters to fit, so you need more data. So it is a time-consuming business, let's say, but it does work. And what we also see is that, and that comes back a bit to the story I told in the middle part. So we again are fighting a bit with this lambda. So if I will just go back to this slide. So it's a bit deciding for 2 things, or you have a lot of droplets with 3 cells or more, and that gives you again a lot of new parameters because you have different interaction parameters. You have higher order interactions playing a role there. So you basically do not want to go too high with your lambda because you don't want to have to include these parameters. But at the same time, that means that if your lambda stays relatively low, you also have a lot of droplets that contain only a single cell. And if single cells can grow by themselves, So if then the signal, let's say, of these single cells that are growing kind of obscures a bit what's happening in the pairwise interactions. So in the system we had now, the single cells couldn't grow. But what we see in the model is that when we allow the single cells to grow, for example, to identify negative interactions. So you have a strain that can grow by itself, but if another guy is present, it cannot grow anymore. So you have an inhibition. But then the model starts to have, it's difficult for the model to accurately predict the parameters because a lot of the kind of signal in your final fractions is now caused by this single cell growth. But we also, again, have ideas on how to do that. We could, for example, try to, in the beginning already, so inoculate the droplets and then make sure that we only take the ones that have 2 cells. So basically get rid of the ones that have 1. But Sagarika's project is also coming to an end, so we don't have time to test that experimentally, but we are testing these kind of questions in the model with simulation data to see. But in principle, it is extendable to larger consortia, but it will need time to do the experiments.
Okay, well, thank you very much, and thanks again for a really nice talk. So
Thank you.
over to Marco. Are you there?